Go for it.
Okay.
Okay, I think your screen share worked. All right. I can see that there are already a bunch of people in the room. Yeah, your screen share is working.
Okay, good.
Yeah. All right. Good morning, everyone. I see that we already have 17 people attending. So it's 9 o'clock on the West Coast. So it's good morning for me, whatever is appropriate for you. I'm going to kick off this session. We have an interesting lineup of 3 speakers. And I think without further ado, I will give it over to our first speaker. His name is Adam Tweed. He is a postdoctoral research scientist in the CALSA laboratory at the Laureate Institute for Brain Research. And he's going to tell us about the relationship between prefrontal cortex and generalized anxiety disorder. So Adam, please take it away.
All right. Yes. Today I'm going to discuss some findings of interceptive abnormalities in general anxiety disorder, or GAD. It is the most common anxiety disorder and is defined by chronic excess anxiety and worry, as well as at least 3 somatic symptoms, including muscle tension, restlessness, fatigue, concentration difficulty, irritability, or headaches. Although these features can accompany autonomic arousal, the DSM de-emphasizes this in GAD by stating symptoms of autonomic hyperarousal are less prominent in GAD than other anxiety disorders. Indeed, studies have shown hyperarousal symptoms reported by GADs did not correlate with autonomic changes. However, no study has examined whether potential autonomic dysfunction in GAD may be better elicited using a causal exogenous manipulation. Thus, we tested whether those with GAD show abnormal physiological, perceptual, or neural responses to cardiorespiratory stimulation via infusions of isoproterenol, an adrenaline analog. As seen in this animation, the effects of bolus infusions of isoproterenol have a 2-minute time course in the healthy population and demonstrate dose-dependent changes in heart rate that allow for the definition of discrete epochs for block fMRI analysis. We focus on the specific epochs, including the dip seen here during the anticipation period after a participant is told the infusion has begun, then the rapid rise toward the peak stimulation, and afterwards the more general, more gradual decline during the early recovery phase. Our approach was a double-blinded, randomized administration of 2 isoproterenol doses, 0.5 and 2 micrograms, and saline given to GADs and healthy comparisons during fMRI scanning. Participants rated cardiorespiratory intensity of sensations throughout the scan by rotating a MRI-compatible dial and then reporting it again post-scan, along with how anxious they felt during the infusions. Here is our consort diagram showing our progression from recruitment assessment of 133 individuals to our final sample of 29 GADs and healthy comparisons. And on the left, some important criteria, or particularly that we excluded panic disorder due to being defined by autonomic hyperarousal, and that we had an all-female sample, as females are twice as likely to experience GAD. The first results I'll show are from linear mixed effects models for the retrospective ratings with dose and group as fixed effects and age and BMI as covariates. We see here a heartbeat intensity ratings show a linear increase with dosage for GAD here in red, revealing a significant difference at the 0.5 microgram dose. They also reported feeling more anxious than healthy comparisons at both doses. Looking now at a heart rate time series illustrating the LME models with the time epochs as fixed effects, we see GAD here in the green lines and healthy comparisons in blue. We see heart rate increases with dosage, as would be expected, but we also find evidence of autonomic hypersensitivity for GAD at the 0.5 microgram dose during the peak of the stimulation, aligning well with the retrospective rating effect specific to that dose. In LMEs for the real-time cardiorespiratory intensity ratings, we saw a similar group difference driven by a 0.5 dose that extended from the peak into the early recovery periods, indicating a heightened interceptive awareness in general anxiety disorder. We then examined potential neural effects related to this autonomic hyperintensity and increased salience by first performing whole brain analysis, for which we corrected for cardiorespiratory related motion while rigorously censoring or dropping participants based on general motion. Contrasts were developed for the isopaternal time course epochs as compared to the baseline epoch and entered into the first level GLM. Group stats were then performed via Afni's 3D t-test++, and I'll be reporting them at a voxel threshold of 0.001, uncorrected, with a cluster level correction of 5% false positive rate. We hypothesized that we would see group differences in insula activation, as we've seen in previous work that has observed isopaternal activates, particularly activates this region. And we do see here robust activation due to expectation effects across all participants during the anticipation period and under saline treatment. As with our prior work, isopaternal elicited more selective insula responses in the ventral insula during peak effect at both doses. However, instead of modulating the insula, we found the novel result of blunted bilateral ventromedial prefrontal cortex, or VMPFC, at 0.5 micrograms during both the peak and early recovery periods in GADs relative to healthy comparisons. We then applied multivariate functional principal components analysis, or MFPCA, to evaluate longitudinal group differences in the overall signal time course rather than according to our experimenter-defined epochs. We summarized scores for the top 2 functional principal components for heart rate, dial, and to help interpret our whole brain result, 3 VMPFC ROIs gleaned from meta-analyses on the relevant functions of self-representation, valence processing, and sympathetic autonomic regulation. MFPCA applies basis functions to the individual raw data, as seen here as the black lines on the spaghetti plot, and models them via a matrix of spline coefficients to characterize the major modes of time series variation around a smoothed mean trajectory, as shown in these yellow fit lines. Here are the longitudinal functional principal component fits for the dial intensity ratings and the heart rate. We saw effects in the LMEs only for functional principal component 1. So here I show those fit lines with GAD again in green and the healthy comparisons in blue for each dose. Differences in the group fit lines reveal results closely matching the epic-based LME results, as GADs were the only ones likely to perceive the 0.5 microgram dose. And their heart rate again showed a hypersensitivity at that same dose. For the VMPFC ROIs, I want to draw your attention to how similar the middle 0.5 microgram pains look across the three ROIs, lending support to our whole brain result. We saw a significant interaction there for group in the self-processing ROI. And for the autonomic regulation ROI, we had a close trend for group at 0.5 micrograms that was significant when I ran LMEs on the raw ROI data with the epic periods as a fixed effect. It just happened to be rather specific to the early recovery period. Finally, the valence ROI is the only one exhibiting differences in each treatment condition, revealing a group effect with no dose interactions. This also held true in the epic-related LME on the raw ROI data. This is important in that the lack of a dose by group interaction here suggests that dose and epic-related effects in these other ROIs were not due to valence effects related to the intensity rating task. So in conclusion, these results provide strong evidence that abnormalities of the autonomic nervous system, in addition to the central nervous system, contribute to the interceptive pathophysiology of GAD. There was evidence of cardiovascular, subjective, and VMPFC neural response differences at 0.5 micrograms, a dose often below conscious awareness in healthy comparisons. These results may indicate failure to adequately constrain autonomic arousal and/or perhaps dysfunctional self-representation of sympathetic arousal, as suggested by our ROI results. We show that the VMPFC is a novel target for understanding and potential treatment of interceptive dysfunction in GAD. Here are my acknowledgements. We're at 12 minutes past, so I'll go ahead and end there and take any questions.
Fantastic. I think you're probably just going to get a round of applause for me because everyone else is muted. It was a great talk. So for the audience, type your questions. Feel free to type your questions into the Q&A, and then use the little thumbs up icon to vote them up. To kick off the questions, what I'm often really interested in when I'm looking at human subject studies is that obviously when you think about arousal, adrenergic response, or anxiety, there are likely to be massive individual differences in how people react even to a shot of adrenaline. Can you guys tease out some of these individual differences? I understand that you guys mostly are looking at group effects, but is it possible to tease out these individual differences, and are they important at all?
I expect that, well, there are, and they are important. It was striking on the group level, how you do have the difference in perception of the interceptive awareness at the 0.5 microgram level. But there is a lot of variation individually, and that would be a good thing to look at that we have not yet, and we don't know. We're coming in pretty specifically at targeting the afferent signal. So once you get into kind of the feedback, feed forward that may propagate, it can be a little hard to know where exactly to attack those individual differences. So that is definitely something to a good forward future direction.
For sure. The other thing I was sort of wondering about, but I think maybe I will let, there's an audience question, so I will just go with that one. There's a question from Lucy Liu. In what way the ROI MFPCA complements the whole brain results from the perspective of the rationale for the PCA methods?
Well, first of all, we wanted to, the VMPFC does a lot of things, and we wanted to parse what the potential functions would be for a reasonable interpretation. As for the MFPCA, we have these experimenter-defined epochs that we were looking at that are based on the healthy comparison, based on healthy population. We didn't know how well that would apply to those with GAD necessarily. So we wanted to kind of take a view that is agnostic of what we had seen before and see how isoproterenol may affect the entire the shape of the time series curve.
Great. All right. Thank you very much. If you wouldn't mind giving your screen share over to Hassan.
Okay.
All right. And so our next speaker is going to be Hassan Aleem, who is a PhD student in the Neurostatics laboratory at Georgetown University, working with Dr. Norberto Grzywacz on how our aesthetic preferences may change over time. Hassan, if you can share your presentation, take it away.
Thank you so much. I had so many screens open, I didn't realize scroll down.
We can see your screen.
So thank you everybody for, and thank you for the introduction. So yes, my name is Hassan, and PhD student at Georgetown University, and today I'm going to be talking to you about something perhaps a little bit different and not familiar to a lot of you, but I'm hoping that it'll be something that garners a lot of interest and questions. So I study aesthetics, and the basic question that I'm going to be talking about today is investigating how these aesthetic preferences change over time. So
Hassan, can I just briefly stop you? Would you mind trying to talk a little bit closer to your microphone?
Sure.
Your audio
Absolutely.
is really weak.
Is this better?
It is a little bit better, yes. Thank you.
So just a quick background into neurosthetics as a field. If you may not be familiar, this is the definition from the Max Planck Institute for Aesthetics, and it basically says that the aim of neurosthetics is to use scientific methods to explain the psychological, neuronal, and social cultural biases of aesthetic perceptions and judgments. So when you read that, you might imagine something like this. You're primarily concerned with looking at a painting, perhaps something abstract, and trying to understand what's going on in the brain. But neurosthetics has become a lot more than just that. It encompasses a wide range of things. For example, questions regarding beauty and all sorts, such as nature, such as food, and the multisensory experiences related to that, dance, fashion, music, architecture, and the list goes on and on. So there's a lot more to neurosthetics and the kind of questions that can be asked, and that's what makes it kind of an interesting field as far as how many topics in the cognitive sciences it kind of dips into. So what are the open questions currently in the field, and specifically the questions that we're studying? So the main questions that we're studying have to do with what is the underlying nature of aesthetic preferences. And this comes in two parts broadly. The first part is, how do aesthetic preferences arise? And to begin to answer this question, we developed a computational model which basically outlines how aesthetic preferences may develop over a lifetime through a reward-based learning process. I'm not going to be talking to you about that too much today, but I'm going to be talking to you about a project that kind of follows that, which basically follows a prediction regarding that model, which is how do aesthetic preferences persist over time. So we know that aesthetic preferences are not stable. Our preferences, our fashions and trends change over time. And when you look across the globe or even across different cities and towns, there's different fashions and trends and preferences. So it's clear that preferences are not stable in any way in time and place. And when you think of the whole developmental life scheme of an individual, they're moving throughout time in these times and places throughout their whole lifetime. So it doesn't, it makes an easy assumption to assume that preferences will not be so stable for the individual overall over a lifetime. But the questions that remain open are, to what extent does this happen and how is this happening? So in an attempt to answer these questions, we, from the current study, which just a few demographical information, had recruited 44 participants through Prolific. And this is the basic outline of the study. We had participants come in for the first visit where they performed 2 alternative forced choice tasks. And this forced choice task basically asked them to pick between 2 different stimuli and in the regards to which they prefer more or less. Then we had them do a brief personality survey / a digit span memory task. And then they repeated this experiment again. So this was our first session. Then subsequently, we had participants come back for another session where they performed the same task with the same stimuli. And this happened a total of 4 more times. So this leads to a total measurement of aesthetic preferences and their stability over a 4-week period. And this is, to our knowledge, one of the only distinct studies that really tries to look at in a fine-grained way what happens to preferences over time over a repeatable weekly interval. So the question that when we were designing the study was, how do we design a study where there's so many different, like I mentioned before, aesthetics span such a broad range of topics. So we wanted to kind of design a paradigm which included a continuous set of context invariant stimuli. And what I mean by that is, so what we did for that was we basically split and used the color space, and we split the color space into a hue, saturation, and value into equally spaced regions, and in 3 categories. And an illustration of that is the following. So we had 3 different categories: shirts, cars, and phone cases. And so as you can see, that this is kind of a continuous space, and this overall stimuli, which is color, is invariant to the different categories, allowing us to see what happens to aesthetic preferences that are bounded and that are changing dependent on different contexts as well. So measuring instability across the different time points is a tough task, because you can measure it in very different ways. And one of the first things that we see in our results is that subjects show a varied individual degree of relative instability in their preferences. And these 2 polar plots illustrate this in a nice way. So just to orient you, the markers basically show the different hue values And the graphs, the actual plots show the amount of times that a certain hue was picked, in this case for t-shirts, in a pre-setting and after 30 minutes. So you can see for this subject, they're showing incredible stability. They're picking something almost exactly the same amount of times, whereas for this subject, after 30 minutes, they're showing some pretty drastic changes in what shirts they picked and what colors they picked in a pre and a post time. So different subjects had different stabilities and across different time points as well. So one method to measure this instability that we used, and I'm going to be talking about here, is to convert these choices into rankings and then use a L1 distance between those to basically simplify how far are the overall rankings between different time points. So that's going to be the measure that I'm talking about in the overall results. So our main result is that aesthetic preferences drift over time. And what you see at the bottom here is the instability on the y-axis and the duration of times on the x-axis. And what you see is that over time, as more time goes on, you see an increase in instability with everything that's not indicated being significant differences. However, a question that was important for us to understand, and this is relative to a starting point. However, a question for us to understand was like, what happens if you freeze one starting point? And here's a little guide down below to make it a little bit easier to understand the different starting points. So A being pre, B being 30 minutes. So we froze that starting point and only looked at what happens if you only look in context to the first starting point. And you see that most of the changes happen within the first week. However, after that, there's a relative stability. And when you think about it, that makes sense because you wouldn't expect in a continuous space for your preferences to completely go in a complete a linear curve and flip completely because they're bounded in a way. So what you see is that relative to your first starting point, you're seeing a change, but then a steady state of instability that is achieved. And when you look at this in terms of all of the time points, what you see is a really interesting pattern developing, which is that successive choices may lead to a sort of crystallization of preferences, albeit with inherent noise. So here you see everything relative to the first time point. And you see that there's a general increase as time goes on, and similarly to the 30-minute and post time point, etc. But however, you do see a downward trend in terms of the baseline stability. So what we believe may be happening is that the mere effects of making a choice and asking subjects to look inward and make choices regarding their preferences would be leading to a concurrent decrease in a crystallization in their preferences and leading to a concurrent relative decrease in stability. However, as time goes on, they're still unstable in general. So that's the noise that exists in the system. When we looked at different factors, we found that there weren't many differences in regards to age, in regards to sex, but we did find differences in age. So as time goes on, or as you look at it across the different age groups, you see that there is a decrease in instability, or basically saying that there is older individuals in our study tended to be more stable as compared to younger individuals. And this is something that is in contrast to a previous finding, which showed opposite results. So it's something that we're looking to investigate a little bit more and understand why the differences may have been arised. So you do see different age and effects as well. So in conclusion, what we show is that aesthetic preferences tend to drift as time passes. And we also see an interesting dynamic interactions where merely making a choice may lead to changes in preferences in themselves. We showed that older adults tended to be more stable, while there were no sex-based differences. And lastly, we also showed that different categories of didn't present this data, but just for audience interest, different categories of the objects did not elicit any differences in stability. And I believe my time is up, so I will.
Hassan, thank you very much for being on time. Your talk has provided a plethora of questions. I think you're able to see the chat. So I would like you to, after this, maybe go into the chat and try to discuss with the attendees there. I'm going to read out a couple of the questions from the Q&A because I'm not sure you can see those. The first one would be a very interesting question. How does one view this in an evolutionary context?
Yeah, so I think it, you can look at it in a few different ways, but for me, I think it makes a lot of sense in an evolutionary context, because if your environment and your body states are constantly changing when you're outside, and when you think about aesthetic preferences or preferences in general, they're one of the main effects is attention and survival. So if your context, is changing, seasons change, etc., or you're not hungry or you are hungry, you would want your preferences to update in regards to the changing environments, and not be, kind of stuck in previous times. Same goes with if you look at different populations. For example, there's studies with symmetry preferences in Hatzatrai versus European populations, and you see that there's a greater preference for symmetry in communities where pathogens are more common. So adapting to the local conditions seems to be the status quo, and something that would make a lot of sense to me.
Great, thank you. Could you actually confirm something real quick? Do you have access to the Q&A panel?
Okay, so I believe I see one question here that says,
right. Yes. So in the interest of time, may I ask you you can type your answer in there. And I think all the attendees will hopefully see that. May I ask you to try to answer that question in the Q&A sort of offline, so that we can move on to our last presenters? Would that be okay?
Yeah.
Fantastic. Thank you very much. Great. And so with this, thank you, Hassan, for a fantastic talk. I would like to move on to our final panelist today, Vivian Tomacek, who is a PhD student in the Budapest Laboratory of Sleep and Cognition, directed by Dr. Peter Simon in the Institute of Psychology at the Eötvös Loránd University in actually my native Budapest. So welcome, hi. Vivian. Please try your screen share.
Okay. Can you see it?
Yes, this is working great. So please take it away.
Fine. Okay, so hello everyone. My name is Vivian Tomacek, and my main interest is nightmare disorder and autonomic activation. Now let's get an idea of this topic by having a closer look at my title: Frequent Nightmares, Parasympathetic Activity, and Heart Rate. First of all, what are nightmares and why are they frequent? Nightmares are vivid dysphoric dreams, including intense negative emotions, usually resulting in awakening, mostly during REM phase with full alertness. Most of us have already experienced nightmares, but it's only considered to be a disorder if it occurs at least on a weekly basis. This REM-related parasomnia has high comorbidity with other pathological states and may be the result of PTSD, but in several cases, the reason behind it is unknown, called idiopathic nightmare disorder, which was the inclusion criterion in this research. The next two parts of the title were parasympathetic activity and heart rate. So let's talk about the autonomic nervous system in a nutshell. Sympathetic activity causes increased heart rate, while parasympathetic activity does the opposite. If the heart rate is higher, its variability is decreased, since the intervals between heartbeats become regular, see the picture on the left, and then the parasympathetic nervous system might take over and slow down the heart rate, and it becomes irregular again, see the picture on the right. And what does it have to do with nightmares? Emotional distress can cause fight or flight response, which is a key component of nightmare disorder, since it includes the intrusions of strong negative emotions such as fear and anxiety, which lead to arousal. Normally, through emotion regulation, we can control our body response, but is this true about nightmare sufferers? Patients with frequent nightmares are thought to have maladaptive coping mechanisms, generating negative effect, activating the sympathetic nervous system, which brings about changes in the heart rate and its variability. HR and HRV can be measured with time and frequency, the main methods, but one question remains. Are we talking about sleep or wakefulness? Well, first of all, we concentrated on different stages of sleep. We based our assumptions on two previous studies. Nielsen and his colleagues found that nightmare dreamers' ratio of low and high frequency of HRV is increased in REM phase after sleep deprivation, which means that the sympathetic nervous system is activated in REM sleep. In the other study, Shimon and his colleagues revealed lower RMSSD, which is the root mean square of successive differences, or in other words, the variance of the length of the intervals between R peaks or heartbeats, and high frequency right before REM phase in pre-REM when the subject's depression scores were controlled for. These results show that lower HRV, along with attenuated parasympathetic regulation, might be common among patients with frequent nightmare. So we hypothesize that nightmare disorder subjects will have lower HRV in pre-REM and post-REM phases. These are transitory sleep stages in the ascending and descending slope of sleep, exhibiting some of the features of REM and non-REM sleep at the same time, and also in REM phase and healthy control subjects, but they will have similar HRV in slow wave sleep, or in other words, stable non-REM stages to healthy control subjects. To further understand the sleep disorder, we aim to explore the differences between the two groups during wakefulness before sleep onset, this is a resting state, and the picture rating task. Our nightmare disorder and control groups had similar distribution of age, gender, and dream recall frequency. Not all the participants' data were analyzed in both parts of the study. For example, there were 24 participants in the nightmare group, out of whom 23 subjects nocturnal and 23 subjects daytime recordings could be used. They reported at least one nightmare a week in the last 6 months. There were 30 participants in the control group who reported less than one nightmare in the last 2 or 3 months. The main criteria to get into any of the groups were the absence of any other comorbid disorders or medical treatment and dream recall at least every second week. With a clinical interview, we excluded anyone showing PTSD symptoms. The recruited participants spent 2 nights in the sleep laboratory, and the second night's recordings were analyzed. The experiment involved several questionnaires, polysomnography during sleep, a picture rating task with electrocardiography or ECG, in which the participants were shown strong emotion-evoking pictures in the second evening as the habitual phase, and in the second morning as the recall phase, and they had to rate them based on how negative or positive they found them and how intense their emotion was. Segments from the ECG recordings of both parts of the research were analyzed, which were 10 minutes long for the nocturnal experiment and approximately 25 minutes long for the picture viewing tasks. In each of the following analyses, I'm going to talk about the mean heart rate separately from the HRV measures. Let's start with the nocturnal segments. Apart from 4 sleep periods, the wakeful period before sleep onset was also included in this repeated measures ANOVA model. In the mean heart rate, significant main effects of condition and group were found, but the interaction was not significant. The post-hoc tests firstly tell us that there is a medium difference between the two groups, and the nightmare group had accelerated heart rate, and secondly, there are significant differences between certain sleep periods, between wake and REM or REM and stable non-REM, for instance, proving a remarkable contrast in heart rate between sleep stages. But what about the wakeful period appearing to be a little different in the plot? Taking a closer look at the sleep stages, we can conclude that in each of them, there is a minimum to large difference between the two groups, and the nightmare participants exhibit elevated heart rate in each sleep period, even during stable non-REM or slow wave sleep, contradicting our assumption. However, they show similar heart rate in the resting wakeful period before sleep onset. With HRV measures, we have controversial results. One more time domain and two frequency domain measures were applied, the RMSSD, the high frequency component, and the ratio of the low and high frequency components. Interestingly, we did not find any significant main effects of group or interaction. Therefore, our results do not show any difference between the nightmare and the control groups in HRV, not even in slow wave sleep, but there is a significant main effect of condition indicating remarkable differences in HRV between sleep stages. The former was quite a surprising result, while the latter is a well-known phenomenon in the literature. As I've already mentioned, to further explore the physiological alterations in nightmare disorder, we added not only the wakeful periods before sleep onset to our analysis, but also the ECG recordings during the picture rating task. The emote refers to the first encounter with the pictures, while the recall part was a shorter task next morning. We found the main effect of conditions significant, with the post-hoc showing that in the emotional habituation part, the HR was moderately higher. The main effect of group was also significant, and the post-hoc comparison demonstrates a little higher HR in the nightmare group, but the interaction was not significant. If we compare the two groups in the two types of tasks separately, we found that during both the first and the second encounter with the photos, the nightmare group had accelerated heart rate. With the HRV measures again, the results are not consistent. We had a significant main effect of condition in one case and a significant main effect of group in two cases, but no significant interaction at all. The significant main effects partly prove the lower HRV in the nightmare group and also points out that the HRV is lower during the emotional habituation task. Some group comparisons also highlight that the nightmare group exhibited attenuated HRV, especially during the emotional habituation task. What do all these results tell us? We found accelerated heart rate in all the sleep stages, that is in pre-REM, REM, and post-REM phases and slow wave sleep, but the HR was also higher during the picture rating tasks, both in the emotional habituation and the recall phases. The faster heartbeats under these circumstances could show that there is less parasympathetic control during sleep and emotionally challenging tasks in nightmare disorder, which can be linked to poor emotion regulation. However, the resting state before sleep onset was characterized by normal heart rate. This is a wakeful state in recumbent position when the subject is quite relaxed and is about to fall asleep. So it is not a surprise that the parasympathetic nervous system can successfully regulate the heart rate in this vigilant but calm period in nightmare disorder. Regarding the heart rate variability, there are controversial results. HRV was normal in all the previously mentioned sleep stages and also during the wakeful period before sleep onset. In fact, the RMSSD, which is also a time domain measure just like HR, was different in the two groups, but still couldn't differentiate them significantly. But the frequency domain measures, the high frequency and the ratio of the low and high frequency were similar in these states. However, with two measures, attenuated HRV was detected during the picture rating tasks, which were used to create emotionally aroused states, and it seems that they did, so we can conclude that strong negative emotions lead to less parasympathetic control. Some questions may arise regarding the normal HRV, like are these results a question of statistical power, or do they mean that if there aren't any dysphoric experiences during sleep, namely they sleep without an actual nightmare, then there is some activation in their autonomic nervous system compared to healthy control subjects, which elevates the heart rate and seems to be universal during sleep. But HRV changes, especially changes in the frequency domain, are connected to the presence of the emotions. Before trying to answer these questions, let me also point out one more important result of this study. We hadn't intended to compare the HRV in the sleep stages, but we couldn't help but notice the remarkable difference between them, which is in line with the well-documented phenomenon that the autonomic nervous system alters in its activation throughout sleep. All in all, our results in connection with heart rate and nightmare disorder are of high importance, since to the best of our knowledge, no other previous studies have concentrated on their relationship. Our results with HRV raise several questions which inspire us to further examine this sleep disorder. First of all, altered HRV might be a trait-like phenomenon of nightmare dreamers, but there has been no conclusive evidence for that. Or it might be state-specific, related to certain negative experiences like emotion-evoking photos or nightmares. However, nightmares are difficult to spot in a sleep lab, so we are planning to examine nightmare sufferers in their own bedroom using ambulatory devices as an attempt to further understand the underlying processes in this pathological state. And these are my sources, and thank you.
Thank you very much, Vivian. I had a quick question. I was wondering if you think that in frequent nightmare patients, the mechanisms underlying the appearance of these nightmares may be similar, or do you think that you're trying to, looking at the variance of your data, would you think that you're trying to tackle a problem that's extremely personalized and very subjective and could be very, the mechanisms behind the nightmares could be very different between patients? I don't know. I don't really know this literature, so I'm curious. What do you guys think? Is this something that is relatively easily generalizable, or is that going to be very difficult?
Well, it's true that there might be individual differences, especially because with nightmares there are a lot of other comorbid disorders, or especially anxiety or depression. So it might cause individual differences, but on the whole, we were looking for trait differences in traits, so trait-like differences, which should be significant if we compare groups or if we compare to healthy control subjects.
Thank you. Thank you very much. And with this, I think I will close this session. I would like to thank all three of our fantastic speakers. I think this turned out to be a very high quality session. I would like to thank our attendees who asked questions and engaged in the chat. And I think last but not least, I would like to thank Elena, our backend, who made all of this possible. All right. Thank you very much, everybody, and have a fantastic rest of your day.
Thank you.
It is being streamed live on YouTube as well. I will post the link here for everybody, for all the participants who are just joining in as attendees. And if there are any tech issues, you can just continue listening to the talk on YouTube as well.
All right, I can see that. Well, we have 3 people so far. We're going to give it a couple of seconds to allow people to join.
Yeah.
All right, and I think we're just going to take it away from here, and people will come. So welcome everybody to, on my time, the 11 o'clock session. I guess it's probably a little bit different for everyone. My name is Yuri Luer. I will be your moderator today, and a little bit of housekeeping. When you have a question, please type it into the Q&A portion of the Zoom webinar. Here you actually have the option to upvote questions. So feel free to use that. We will try to keep an eye on the chat as well. So if you happen to want to share something in the chat, then feel free to do so. And then without further ado, I would like to introduce our first speaker, who's going to be Inok Zhang, who is a PhD student in Dr. Sungmin Cho's laboratory at Johns Hopkins, and who will tell us about new quantitative methods assessing EEG. Go ahead, take it away.
Okay, let me share my screen. Okay. So very happy to have you all to listen to my talk on a novel quantitative method to assess EEG reactivity in comatose patients. So a coma is a prolonged state of unconsciousness. So during a coma, a person is unresponsive to external environments. Assessing the consciousness, or say the responsiveness of those patients, is important for the clinician to guide the clinical treatment. And the current clinical practice is to use behavioral tests, such as like the Glasgow Coma Scale, which use the eye-opening, motor response, and verbal response to assess the responsiveness of those patients. But as you can see, this will vary by the skill of the performer, and it's qualitative rather than quantitative. And there's a misdiagnosis rate, which is reported to be high. So we want a quantitative method, or say an objective method, to help the clinician to assess the consciousness of those patients. Here comes EEG reactivity. EEG reactivity is defined as a change in cerebral EEG activity due to external stimulation. Here I show some example. So you can see several EEG channels, and here's a stimulation, which is a loud noise of a clap. After the clap, you can see some EEG channels show a different pattern compared before the stimulation is induced. And you can see some band power or spectral power change in the delta and theta frequency. And this is the EEG reactivity. EEG reactivity has been shown to be associated with outcomes in comatose patients. For example, in a study conducted by Logi et al., they showed that patients who showed EEG reactivity has a higher rate to recover consciousness compared with those patients who did not show EEG reactivity. So although EEG reactivity is getting attention, the patterns of EEG reactivity exhibited by those comatose patients is not clearly studied, such as how EEG reactivity differ in response to different type of stimulus, how long does the reactivity last, and so on. So here in this study, we analyzed the EEG data from comatose patients on ECMO, and we applied 3 types of stimulations: auditory stimulation, noxious stimulation, noxious touch stimulation, which is the nail bed pressure pinch, and nostril stimulation. We aim to analyze the characterization of the EEG reactivity in those comatose patients, especially on the types of EEG reactivity they exhibited, the time, the start time of the EEG reactivity, the duration, and how does the reactivity differ in different brain regions. And finally, we want to design a scoring method to evaluate the EEG reactivity in different patients. Here is the study flowchart. Basically, we include the comatose patient on ECMO, and we apply the 3 types of stimulation. After filtering for the artifacts and noises, we have 24 patients data in our analysis. We separately apply the quantitative EEG analysis and a visual analysis by the EEG expert, which is our reference method. We first study the correlation of EEG reactivity, and then we compare the calculated reactivity score with the reference method. Here is a more detailed methodology. So I want to make the long story short. We basically have 2 windows, which is 10 seconds before the stimulation and 10 seconds after the stimulation. For each time point inside this window, we calculated the band power, or say spectral power, and permutation entropy for each EEG channels. And for each measured value, because we have 19 EEG channels, so we use the average value among 19 EEG channels. Here is an example of how does the measured value change over time. So you can see the black flat line, which is the baseline, and then you can see the value change higher than more than 2 standard deviation of the pre-stimulation period, and then drop below minus 1.96 or say two standard deviation. So we first perform a standardization for this value, and then how do we calculate score? We first take the absolute value of the post-stimulation period, which looks like this, and then we only take the value that's higher than 1.96, which is our criterion for reactivity, and then we take the mean of this value. We sum all the means from different metrics of the measurements, which is the final reactivity score. Okay, maybe you can figure out, you can see that the EEG change can either increase or decrease, right? So we define two kinds of EEG reactivity. One is the increased EEG reactivity, which is the measured value increased higher than the 1.96 standard deviation of the pre-stimulation period, which I would say refer to the criterion right after the stimulation. And another is the decreased EEG reactivity. The measured value decreased below the criterion after the stimulation. And you can also see that this value would not just only go up. It can go up and then go down, or it can go down and then go up. So in this case, it goes up more than the criterion and then drop down more than the criterion, or the opposite way. So this will be classified as a both increased and decreased EEG reactivity. So we define 3 types of reactivity. So this is how does these different types of EEG reactivity distribute that we in different stimulation and different metrics we calculated. So the y-axis are the percentage of the EEG reactivity we detected in all those EEG epochs, and the x-axis are different measurements: spectral power in 5 frequency and permutation entropy in 5 frequencies. Blue is increased EEG reactivity, and the orange is decreased EEG reactivity, and the yellow is the both increase and decrease. You can see that the increased EEG reactivity is most common in the spectral power across all 3 different types of stimulation. While in permutation entropy, the decreased EEG reactivity is the most common in auditory stimulation, but the increased is the most common in permutation entropy in auditory stimulation, but decreased EEG reactivity is the most common in permutation entropy in noxious touch and nostril stimulation. And the both increase and decrease are the less common here. Okay, then we analyze the start time of the EEG reactivity. The start time is defined as the value, the measured value change, or say first hits the criterion. And here is the results. We found that more than 50% of the EEG reactivity happened within 4 seconds after the stimulation, and more than 90% of them happened within 8 seconds of the stimulation. And due to the limitation of our time window, which is only 10 seconds, we don't know if the EEG reactivity can happen after 10 seconds or not. And here is the results of our analysis on the duration of the EEG reactivity. And we found that the duration of 2 seconds was the most common, while the other duration appeared with a very similar frequency. Again, because the limitation of our time window is only 10 seconds, we don't know if the EEG reactivity can last for more than 10 seconds or not. So here we analyzed the topographic maps, or say, how does the EEG reactivity differ in different brain regions? As you may remember, we are using the average value of all 19 EEG electrodes to calculate or say to compute the EEG reactivity. However, in some cases, this does not work. Here is an example. If this is a brain and some brain region actually increased while the other brain region decreased. So this, in this case, if you use the average value of all 19 EEG electrodes, increase and decrease will actually neutralize. So you would not detect any EEG reactivity in this case. So we define a term which is we call the disagreement. Okay, so disagreement is the number of sites that, or say the electrodes that had the opposite EEG reactivity compared with the average of all EEG electrodes. For example, here, the average is actually decreased. However, 5 sites of electrodes are increased. So in this case, we will say that this agreement is 5 sites. Here's the results. We found that more than 60% of the EEG reactivity detected doesn't have any disagreements, and more than 95% of the EEG reactivity we detected only have 3 or less sites of disagreements. In general, these results support that we can use the average of all electrodes to measure the EEG reactivity. So finally, I'd like to compare the reactivity scores that's calculated by our quantitative analysis with our reference method, visual analysis of EEG reactivity by the EEG experts. The visual analysis will categorize the EEG epoch for each patient into present or absent of EEG reactivity. And this is a very common practice in the clinical environment. And then this graph shows that the y-axis is the reactivity score, and x-axis are different stimulation and different groups. Present is the blue, and the yellow is the absence. This is categorized by the visual analysis. And the t-test told us that the present group have a significantly high EEG reactivity score compared with the absence. And this is significantly high within all 3 stimulation. Okay, in conclusion, we analyzed some characterizations of EEG reactivity in comatose patients on ECMO, and our findings provide preliminary support for the validity of the EEG scoring method we used. However, this is only done in a very small patient population, and a further validation of larger population is needed. And this is all about my presentation. Thank you.
Thank you, Enoch. I think definitely deserve, unfortunately, a silent applause because of the webinar format. We definitely have time for a question or two, and I would like to kick off the questions with asking, when you're looking at, so you're looking at these sort of evoked changes in EEG in comatose patients. Can you comment, for someone who's not in this field, can you comment a little bit on how these evoked changes would look like in a non-comatose patient? How does what you're recording, how does it compare to, say, a calm normal patient or a normal person, say with an auditory or a noxious stimulus?
Okay, so unfortunately, we didn't conduct the same, we didn't use the same method on a healthy subjects. So we currently don't know what does this method would look like when it applies to a healthy subject. However, there's some other study that is conducted in this similar field on analyzing how does the EEG reactivity changed in either different patients with different disease or in a healthy subjects. And interestingly, they will have very similar thing. Like here I show a topographic map. You can see some brain region will actually light up, and while other brain region will remain the same, doesn't have activity, right? So the healthy healthy subject will also have the same pattern, but the amplitude will much higher than those in comatose states. So this is a very main difference between comatose and healthy subjects.
Great, thank you very much. I would like to move on to our next speaker in the session, Ai-Fong Tong, who is a medical student at the University of Washington. She also participated in the 2019 cohort of the NIH Medical Research Program, and she will tell us about cortical ripples in humans. Please take it away.
Thank you for that introduction, and thank you everyone for listening in. So I will talk about our study on ripples in human cortex and how it reflects the spectrum of synchronous spiking activity across spatial scale. So first, I want to start off with a framework for understanding the mechanism for cognition, which we can think of as these transitions between asynchronous and synchronous activity, whereby at one end, you have neurons that represent different information and have distinct spiking. And then there are periods in which neurons that have shared representations communicate by spike synchronization. And these perform computations that are necessary for cognition. So we study oscillations in the brain from local field potential recordings and from intracranial EEG recordings in order to assess the level of synchronous activity in the brain that is important for cognitive processes. So in particular, we've looked at 70 to 200 hertz frequency band, which I will refer to as the high frequency activity. And we've seen that we can detect synchronous activity that it underlies attention and successful memory retrieval and encoding. And then more recently, we've also found that the 80 to 120 hertz band, in particular in humans, which I will refer to as the ripple band, is also important in successful memory retrieval. And we know from vast rodent literature that these ripples can be detected as events of synchronous activity. And similarly, we've done this in humans, but we use these arbitrary detection thresholds, and it's still unclear whether these ripple events can represent the synchronous activity in this spectrum of synchronous activity. And also we measure this ripple signal in macro scale recordings, and it's unclear if these macro scale recordings at the ripple band actually represent this spectrum of synchronous activity. So we explore the ripple signals in human cortex further. In 21 patients with intracranial EEG recordings, we asked to what extent do ripples resemble high frequency activity at the macro scale? And in 6 participants with simultaneous intracranial EEG and local field potential recordings, we asked how are ripples and spiking activity related across spatial scale? And we asked to what extent are synchronous spiking activity and ripples rhythmically modulated across spatial scale. So we measure local field potentials from microelectrode arrays, and we can compare these microscale signals to macroscale signals from intracranial EEG contexts nearby. And so during these recordings, we asked the patients to participate in a paired associates verbal memory task in which there are 2 periods. The first encoding period, we cue the patient to form a memory when they're presented with a word pair from a list of word pairs. And then we have an interval period in which we give math distractors, and we follow that with a retrieval period in which the patients are cued to recall a memory when they are presented randomly with 1 word from the previously studied word pairs. And then we take the recordings offline and we can assess the ripples and the high frequency activity at the macro scale and at the micro scale and compare it to underlying spiking activity. And we can assess how these signals are related during memory retrieval. And so first, in order to compare ripples and high frequency activity, we decompose the intracranial EEG signal using a wavelet transform. And from the time frequency plots as shown on the bottom, we observe these discrete periods in which there's high activity at the high frequency range. And specifically, there seems to be high activity in the 80 to 120 hertz range, or the ripple band. And we can use the ripple detection thresholds that we previously published on to identify these discrete narrowband events. And so we can compare the high frequency activity and the rate of ripples prior to vocalization during retrieval, as indicated by the time 0. And from the time frequency plot on the left, we can see that there's an increase in high frequency activity just before vocalization across all trials. And we can similarly see an increase in the rate of ripples just before vocalization, as seen in the raster plot underneath. And when we separate out correct trials and incorrect trials, we see that specifically successful memory retrieval exhibits increased high frequency activity and increased rate of ripples. And further, when we compare the change in high frequency activity and relate it to the change in ripple activity, we find that these measures are correlated in memory relevant brain regions. So specifically in the medial temporal lobe and in the temporal association cortex, or the anterior temporal lobe. And so these findings suggest that ripples are a measure of synchronous activity that's represented by high frequency activity changes resolved in time. And then next, we wanted to know within temporal association cortex, how are signals across spatial scale related? And so we make pairwise comparisons across spatial scale, and we modified the ripple detection threshold in order to assess smaller events. And so first we compared spiking activity and microscale ripples, and then spiking activity and macroscale ripples. And just from looking at ripples across spatial scale, we see that when these ripples occur, we also have more spiking events occur. And so when we computed the correlation between ripple amplitude and spike rate, we found a significant correlation for both the microscale and macroscale across patients. And this is demonstrated in the middle plot, where each data point represents the correlation for one patient. We similarly looked at individual macroscale ripple events and characterized the spike content for each ripple event by looking at the proportion of spiking units. And we found that macroscale ripples reflected the number of spiking units. We further compared macroscale ripples and microscale ripples. So this time we just focused in on the ripples. And so from looking at the signals simultaneously, we found that when there were more microscale ripples, the macroscale ripple tended to be larger. And so when we computed the correlation between the macroscale ripple amplitude and the proportion of microelectrodes with ripples, we found a significant correlation, suggesting that macroscale ripples reflect the sum of microscale ripples. And then we further observed that when the microscale ripples were more aligned, these were associated with larger macroscale ripples. So we wanted to measure the synchrony of microscale ripples using pairwise phase consistency. And so to the right is a diagram showing how we assess synchrony of microscale ripples using these pairwise phase differences across the ripple signals in the microelectrodes. So from that analysis, we found that macroscale ripples not only reflect the sum of microscale ripples, but also their synchrony or alignment. And so further, we looked at the relation between spiking activity and microscale ripples. So this time we assessed the LFP or microscale ripple amplitude correlation with the spike synchrony as assessed by, again, the pairwise phase consistency, and found a significant correlation between these measures, suggesting that microscale ripples reflect the synchrony of spiking activity. So altogether, this suggested that macroscale ripples reflect synchronous microscale ripples, and microscale ripples reflect synchronous spiking activity. And then next, we wanted to know to what extent are synchronous ripple and spiking activity rhythmically modulated. And so we computed the spike triggered average as shown on the left top and the ripple triggered average as shown on the left bottom. And we found that these events appeared to be coherent with low frequency oscillations. When we computed the pairwise phase consistency of these events across frequencies from 2 Hz to 200 Hz, we found that the spiking activity and ripples are modulated by low frequencies around 3 Hz. And so further, we wanted to know now with synchronous spiking activity and ripples, so events that occurred coincident with ripples, how were these activity modulated? And so again, we computed the pairwise phase consistency for these events during ripples, and we found that they were modulated by low frequencies at 6 and 11 Hertz. And so altogether, this suggests that both ripples and spiking activity across spatial scale are modulated by low frequency oscillations. And so again, to come back to our framework for understanding the mechanism for cognition, we can think about ripples as these periods of rhythmically coordinated communication that occur during these continuous periods of distributed computations. And so when we detect ripples, we indeed can detect these periods of synchronous activity along the spectrum of activity. And specifically, when we detect larger ripples, that may indicate more shared representations across neurons. And so overall, our findings would suggest that these ripples measured at both the macro scale in the intracranial EEG recordings and the micro scale in the local field potential recordings reflect transient engagement of synchronized spiking activity during successful memory retrieval. With that, I'd like to thank my lab, Dr. Zaghloul's lab, and I've left my contact information if anyone has questions offline. Thank you.
Awesome, thank you. So I'm by no means an expert in this field, but my understanding from the rodent literature is that during an event, there's some spiking activity that happens in a neuronal ensemble, and then during a ripple, this same spiking activity is often sort of replayed, you know, to enforce these memory events. Since you guys are actually recording spiking activity in humans, which I have to say is absolutely phenomenal, do you guys see a pattern like this? Can you tease something like this out in the human data?
Yes, so there has been a paper published by another member of the lab that has found that there is a reinstatement of these spiking events or the sequence of spikes, and that they were correlated with these repeats in the ripples that occurred during encoding and also similarly occurred during retrieval.
That's awesome. That's really cool work.
Thank you for the question.
All right. Thank you very much. I think in the interest of time, we will move on to our next speaker. I would like to ask all of our panelists to keep an eye on the chat in case someone asks a question in the chat. Oh, actually, I think we have time. There's a quick question in the Q&A to you, to the previous speaker. So I might try to quickly add that in. The question is there any trace that the ripples can be subject of plasticity?
Yes, so since we're finding that ripples may represent this synchronous activity, typically we can think of synchronous activity as these consistent patterns of spiking events. And so when you have one spike occurring consistently after another, this can generate stronger connections or more and increasingly plastic connections, if that makes sense.
Fantastic, thank you. If you guys can keep an eye on the Q&A, you actually have an option to answer the question in writing in the Q&A. So, you know, I see that there was a new question popping up. I think in the interest of time, we will move on to our last speaker, but please try to engage with the audience through the Q&A panel. All right. And so our final speaker in this session is Mónica González Garcia. He's a PhD student in the Department of Psychology in the University of New Mexico, and she's working with Dr. Derek Hamilton, and will tell us about the effects of moderate prenatal alcohol exposure. Take it away, Mónica.
Okay, thank you so much for the introduction. So I'm a second-year grad student, and what I'm going to be talking about is a pilot study that we started developing in our lab. So I'm interested in the effects of prenatal alcohol exposure on LTP and rhythmic activity in the dentate gyrus. According to the CDC, to give you some background, the prevalence of drinking during pregnancy is about 1 in each 9 pregnant women reported drinking alcohol in the past 30 days. And from those, about 1/3 engaged in binge drinking with an average of 4.5 binge drinking episodes. So the result of being exposed to alcohol during pregnancy is known as fetal alcohol spectrum disorders. And there are several disorders that can be considered FASD. One of them is FAS, or fetal alcohol syndrome, and that affects, and this is data only for the US, from 0.2 to 1.5 children for every 1,000 live births. And it costs about $4 billion each year. So one of the implications in terms of neurodevelopment of the FAS are cognitive deficits that can persist throughout the entire life of the individual. And among the several domains that's involved, we have learning and memory, which is what my interest. So we don't know exactly what the mechanisms and the neurobiology contributing to those deficits are. And so because of that, we don't have effective treatments. So the broad aim of this study is to better understand the effects of prenatal alcohol exposure, that is, I'm going to refer as to PAE, on long-term potentiation, or LTP, and hippocampal function using in vivo electrophysiology. So I'm using an animal model, and here, let me try to use my pointer. Here I have a comparison between the human brain and the rat brain. And so the reason why you're using an animal model is because of the anatomical and physiological similarities with humans. So the hippocampus is the structure in the limbic system that is involved in learning and memory, and it's highly susceptible to alcohol during prenatal development. So this cartoon here shows like a slice of the rat brain showing the regions of the hippocampus. So a mechanism that's involved in learning and memory, which is known as LTP, refers to the strengthening of the synaptic connections. And LTP can be found in several synaptic pathways all over the brain, but my focus is on the dentate gyrus because models of alcohol exposure have yielded consistent alterations in the dentate gyrus, but not in other pathways. One of the possibilities is because the dentate gyrus is the primary cortical input from the interneural cortex into the hippocampus. So here at the bottom is just a representation of what is a normal neurotransmitter release with some receptors here at the postsynaptic terminal. And then after we do high frequency stimulation, we can have an increase depending on which phase of the LTP we're talking to. We can have more neurotransmitter release or also increase in postsynaptic receptors. So here are the regions and pathways of the hippocampus, and I'm interested in this outline portion down here, which is like the lateral perforant pathway and the medial perforant pathway. And so are the places where I put my electrode, and I'm using a monopolar electrode. So I have a recording in the hilus of the dentate gyrus and another monopolar electrode in the perforant pathway. This is like the stimulating electrode right here. And here is a simplified diagram of the local interneuron feedback circuit and showing the inputs from the perforant pathway onto the granule cells, basket cells, and so the red arrows are showing the direction of the excitatory synapses, and then the blue ones showing the inhibitory synapses. So basically like the granule cells sending inputs to the basket cells and the interneurons, and then receiving the feedback from those cells. So in my recordings is an example of how the response looks like. So here at the bottom, I'm recording the continuous EEG. So we have a normal activity here, and it's only one channel. And here in this red square or rectangle, here is the evoked response. And so on the left, we have the overall shape of the response of the field potential or the field PSP without the pop spike, which represent the inputs coming into the cell. And here on the right side, it's the same response with the pop spike. Which is like when the granule cells, like the population spike is when the granule cells become active, and then that activity gets subtracted from the field on this portion right here. So basically what I'm showing here is how, when I do the analysis, which parts of the response I'm looking for. So I'm doing the analysis of the slope of the population spike, and also this wavy portion right here, which is a later portion of the response, which is related to the feedback circuit. And also sorry, I'll go to the next one. Right, so for this one, because I'm doing time frequency analysis, so it's the same, this is one of my real data. It's just showing the red line right here shows the stimulus when it occurs at 0. And we can see how we have this, in the time frequency analysis, we have more power associated with the early portions of the response right here. So I'm gonna talk about this later. So the specific aims of this study is how does PAE affect LTP, the dentate gyrus, and how does PAE influence continuous EEG signaling and time frequency relationships in evoked response after high frequency stimulation. So for my methods, we have a drinking paradigm. It's a voluntary drinking paradigm, meaning that we're not force feeding the animals with alcohol. They're exposed to, they have during 4 hours, 2 weeks before breeding, then they have breeding and gestational period. They have alcohol available during 4 hours a day, and the amount they consume is equivalent to 0.086%, which is it's close to the legal driving limit in the US, or equivalent to a few glasses of wine. So I used 7 saccharine animal and 6 PAE. I'm using urethane anesthesia. And I already talked about like where I'm putting like the recording and the stimulating electrode. So we record for 20 minutes baseline just to make sure that we have a stable baseline before doing the high frequency stimulation. Then I do 5 trains of high frequency stimulation for 12.5 milliseconds. And after that, we record for 60 minutes the evoked response at we record one at each 30 seconds. And I'm using the EC40 protocol, which is 40% of the maximum evoked response that we get. So, and here, it's an example of one of my animals. This is baseline, and you can see the slope here and the pop spike, how it was, and then the arrow represents the high frequency stimulation and how it looks like after. So this is the overlay of the entire session, like 20 minutes of baseline and 1 hour of post HFS right here. It's hard to see, but we can see that the slope became steeper after high frequency stimulation than it was before. So it means that we have more cell depolarizing right here. And of course we can see like it's clear how it increased like the slope, the pop spike. So here, the results show no difference for baseline for both groups. And we see that LTP did occur for both the PAE, which this line down here, and the saccharin. The big difference that we see here is that even though we had evidence of LTP occurring for both groups of animals, we can see that we see more change for the slope for the saccharin animals than the PAE, and also it's more stable. We have more sustained potentiation over time for the saccharin than we have for the PAE, where we see more decay over time. For the power analysis, which is how much amplitude of the individual signal frequencies is represented in the signal, we have a comparison for baseline and post-HFS, blue representing the saccharin animals, and yellow the PAE. So we have more power from the baseline and then for post-HFS for both for the PAE compared to the saccharin animals. For the time frequency, so this is the plot that I showed you before, because I was decomposing the response to see what was happening in early portions of the response right after stimulation and later in that wavy portion that part of the response that I talked about before. So what we're doing here, I had like a bigger matrix, and here's only the last line that I'm showing. This is the difference for baseline is the PAE animals minus saccharin. So the blue means the greater power in saccharin animals. So we see more power associated with early portions of the response. So here's right after the high frequency, right after the stimulus. So it was more power for the saccharin animal than the PAE. Then after post-HFS associated again, right after the stimulus, we see it's less evident, but it's still more power for the saccharin animal. And then this last plot here is the interaction. So post-HFS, PAE minus saccharin minus the baseline PAE minus saccharin. So what we see here is that right after the stimulus, we see a little bit more power for the saccharin, but in later portions of the response, we see that the PAE animals, they are maintaining more power. So what this suggests is that there is some disruption in the feedback circuit, because we know this part is associated with the interneurons activity. So the summary is that the PAE animals exhibited less potentiation of the field EPSP and LTP compared to SAC animals. The time frequency analysis suggests that the PAE maintained more power in later portions of the evoked response that could be due to disruption in feedback signaling from local interneurons. And the broader conclusion of this study is that there is an alteration in the function of interneurons that's evident on the potentiated response that could have functional consequences. So as future direction, I'm gonna do a recording at subthreshold EC20. And the reason for that is to isolate the feedback signal in the time frequency analysis. One of the possibilities of the interneuron dysfunction is that if there is something there that could be a potential target for pharmacological interventions. So, and this study is part of a larger study going on in our laboratory that is investigating some anti-inflammatory PPAR gamma agonists on PAE. And also we are studying some experimental histaminergic drugs on LTP of, exposed animals. So I'd like to thank you, my contributors, which is my mentor, my secondary mentor, Dr. Savage and Susie Davids. So thank you so much.
Thank you, Monica. We may have time for a couple of quick questions. And to kick off the Q&A, I was wondering, you showed us the LTP recordings over the first hour, and you have very nicely showed that the initial decay in PAE animals is much faster than in normal rats. But then both seem to sort of level out to a steady state. I'm curious whether you guys have done longer recordings to see if in the PAE animals, the LTP actually decays back to baseline level earlier than in the controls, or whether there is a smaller amount of LTP on the long run, but the total decay would be similar. I didn't realize it would require much longer recordings. Have you guys attempted anything like this?
Yeah, so for some of our data that is not shown here, we did do a longer session, which was twice as long as the one I'm showing here. That was just an hour. So we have done some recordings we had like 1 hour of recording, then we had a wait period, and then we had another hour just to see how it was changing. So we continue to see some decay. Some animals was a little bit returning to baseline, but not all of them. So what we saw is that some of the PAE animals, they looked normal or compared to the saccharine. So everything was behaving the same way. And some other animals, they would go back to baseline. But we did see some controls as well that were kind of not exactly going back to baseline, but toward baseline. But we didn't do any recording longer than I would say 3 hours. Just see that. Thanks for the question.
Great. Thank you. I think just because we are sort of running out of time and I think we will be kicked out of this Zoom room, I would like to finish off with thanking all 3 of our speakers. I think this was a fantastic and very informative session. I would like to give a quick shout out to our backend, Taha, who made the technical parts of this session possible. And I hope all of you guys will have a fantastic rest of your day.
Thank you, everybody.
Thank you. Thank you, everyone.
Enjoy your days or nights, wherever you are.
Technical side of things, which is really terrific. So Gary Somasekar is going to help us do the technical side today. And we have 3 very interesting presentations relating to rhythmic activity in the central nervous system, in the human central nervous system. And of course, I get excited about that because that's what I do. So I'm actually looking forward to seeing these presentations today. And they cover various aspects of brain function as well in adults and children. And there is also intracranial activity. So there's something for everyone here. So without further ado, I'm going to introduce Lisa Johnson as our first speaker, and she's going to talk about some slow theta activity in visual scenes in a memory task. And so, Lisa, the floor is yours, and we will start timing. And you are muted, Lisa. Yep.
All right, thank you for having me. I'm a postdoc in the Night Lab at UC Berkeley, and today I will present some brand new results from a collaboration with Noah Ofen and colleagues at Wayne State University. For the last few years, we have developed a pediatric intracranial program and published the first studies using direct brain recordings to look at the development of memory in kids. And my colleague Shin will actually give a talk later in this session. So first, a little background. Neural oscillations are ubiquitous, with tens of thousands of papers to date associating them with everything from physiology to pathology to cognition. Oscillations have been studied non-invasively using Scalp Eeg for about 90 years. To study neural oscillations, we break down the EEG signal into waves of different frequencies. Frequencies, measured in hertz, refer to the number of cycles in 1 second. Frequency band-limited oscillations reflect general different rhythmic fluctuations in neuronal excitability. They are generally associated with different functions. As I'll focus on today, oscillations in the theta band are widely associated with memory and cognition more broadly in humans, as well as animal models. Theta oscillations provide optimal windows for neuronal firing and optimal windows for interactions between brain regions. Neural oscillations change considerably during development. This makes understanding their development fundamental to understanding the development of the brain. For instance, since the first human EEG in the 1930s, researchers have focused on alpha oscillations. These oscillations dominate the EEG signal at rest. As shown on the left, in a study from 1939, alpha oscillations emerge during infancy and exhibit rhythmic waveform patterns within a few months. As shown on the right, these rhythms reach the canonical 8 to 12 hertz range, or adult-like levels, around age 12. Scalp Eeg is a wonderful tool, but notoriously limited in spatial resolution. It does not access medial regions directly and offers imprecise measures of the lateral regions it does access. It therefore offers a limited window into the development of cognitive processes such as memory, which may rely on the medial temporal lobe and its connections to specific regions of the cortex, here prefrontal cortex. So brief overview, declarative episodic memory is what we usually think of as memory. The medial temporal lobe interacts with the prefrontal cortex during the formation and retrieval of memories of events in our own lives. Although kids as young as 4, 5, and 6 years of age do form memories of events in their own lives, it is widely understood that memory continues to undergo improvements through adolescence and even into young adulthood. These improvements co-occur with the maturation of medial temporal prefrontal networks. Specifically, the protracted development of memory into young adulthood parallels the protracted structural maturation of the cortex, as shown on the left, and long-range white matter tracts connecting the medial temporal lobe to different regions of the cortex, as shown on the right. And in this box, I just highlight the age range from about age 5 to 25, indicating protracted structural maturation into young adulthood. So, to access the medial temporal lobe and prefrontal cortex, we capitalize on the opportunity to record EEG activity intracranially, directly from the brains of children and adolescents as they perform memory tasks. These electrodes are implanted invasively for the management of medication-resistant seizures. If seizures are suspected to have a focal source, patients are implanted with electrodes subdurally, as shown on top, or stereotactically into deeper brain structures, as shown on the bottom. Then, these patients spend about a week in the hospital for intracranial monitoring. At the end of the week, they get the electrodes removed, and if all goes well, the seizure focus resected. During this time, we show up with a pop-up lab, run memory tasks, and copy the clinical data. As you can imagine, these unique data provide unprecedented access to the human brain. They are high not only in temporal resolution, but also spatial resolution. And with no skull, skin, or hair in the way, they provide sufficiently high signal-to-noise ratio to study memory in single trials. The application of these clinical data using ECOG and SEEG to study memory, as well as cognitive neuroscience more broadly, has risen pretty dramatically in popularity over the last several years, and for good reason. To extract theta oscillations, we first decompose the power spectrum into different frequencies. We can then apply algorithms that separate the periodic or oscillatory signal from the aperiodic or 1/f signal. The 1/f signal in this graph in red is estimated and subtracted from the true signal in blue, leaving just the bumps, which reflect band-limited oscillations, or are purported to. As shown in this figure, the black trace, which is the true signal minus the 1/f non-oscillatory signal, shows a bump at about 10 hertz, reflecting the well-known alpha rhythm. In human adults, researchers have used this technique to isolate theta oscillations in the medial temporal lobe during memory and related cognitive tasks. These particular data from a virtual navigation task. 2 studies published very recently this year have reported the existence of multiple theta oscillations in the adult hippocampus at both ends of the canonical theta band. One is a slower oscillation around 2 hertz, and the other is a faster oscillation around 8 hertz. These oscillations may be functionally dissociable during memory and related cognitive tasks. In our work, we use a tried and true subsequent memory paradigm to look at the development of memory in kids and adolescents. In this task, images of scenes are presented during a study phase. During this phase, participants indicate whether each scene is indoors or outdoors. This is not a test, but promotes depth of encoding and ensures that we, as researchers, know which scenes were actually attended to at study. Participants are then tested on all the scenes at study, intermixed with foils and a recognition memory test. We analyze the data during the presentation of each scene during the study phase. These data can then be analyzed for all attended scenes, which is over 90% here, and as a function of whether the scene was remembered or forgotten at the test. This is a widely used approach to isolating the functional substrates of memory formation. So far, we've tested over 40 children and adolescents intracranially on this task. On the left are the coverage maps of 2 example patients from our cohort. Typically, these subdural ECOG grids cover lateral prefrontal cortex as well as cortical sites in the medial temporal lobe. On the right, critically, I demonstrate that patient behavior shows the expected developmental trajectory of improvements with age. Memory outcomes improved here from age 5 to 25, and the slope is identical to that of a large cohort of non-clinical controls. Patients are highlighted in red. This particular study has 2 overarching aims. First, describe the developmental neurophysiology of medial temporal prefrontal networks. Second, relate this developmental neurophysiology to the development of memory. To address these aims, we selected 21 children and adolescents from the larger pool based entirely on coverage of our regions of interest, the medial temporal lobe and prefrontal cortex. Across the whole group, we were able to isolate 80 medial temporal and 357 electrodes in PFC. Prefrontal coverage was further divided into subregions based on individual anatomy into inferior, middle, and superior frontal gyri. Focusing here on the first aim, I isolated theta oscillations during the study phase of all attended scenes. After subtracting the 1/f aperiodic estimate from the power spectrum, I isolated multiple theta oscillations in the medial temporal lobe of these children and adolescents. Consistent with reports in adults, I detected a slower oscillation just below 3 Hz and a faster oscillation just below 7 Hz. Each of these plots shows the group results aligned to the individual peaks, slow and fast theta frequencies, respectively. Almost all channels exhibited a faster oscillation, and about 80% of channels also exhibited a slower oscillation, consistent with adults. Furthermore, I found the same pattern throughout the prefrontal cortex. I detected a slower oscillation just below 3 Hz and a faster oscillation around 7 Hz. And just like the medial temporal lobe, almost all channels exhibited both a faster oscillation, and about 80% also exhibited a slower oscillation. Finally, I observed that both theta oscillations shifted with age. Specifically, faster oscillations were faster and slower oscillations were slower over the 15-year age range studied here. This double dissociation of frequency by age was observed in the medial temporal lobe and in all 3 regions of the prefrontal cortex. To summarize, we observed oscillations at both ends of the canonical theta band, consistent with recent findings in the adult medial temporal lobe. Both fast and slow theta oscillations were observed not only in the medial temporal lobe, but also the prefrontal cortex. Both frequencies shifted with age in a double dissociation in all regions studied, linking neural development to the functional differentiation of the theta band. Further analyses indicated that both theta oscillations support medial temporal prefrontal functional connectivity patterns, a topic for another talk, that were predictive of subsequent memory formation. So this result critically links not just the fact that we found these oscillations, but links them to behavior and behavioral gains over development. And taken all together, these findings suggest that the functional differentiation of the theta band may be a core component of the development of cognitive processes such as memory, which rely on medial temporal lobe, prefrontal cortex, and the connections between these 2 regions. Thank you for listening. Thank you especially to the kids and their families who let us come into their hospital rooms and record their brains. Thank you to my collaborators at Wayne State and broadly throughout the University of California. As you can imagine, this work takes a lot of multi-institution collaboration with doctors and researchers. And thank you to the funding agencies. And thank you for listening.
Thank you, Lisa. And we don't have any questions right now that I can see, but that gives me the opportunity to ask you a question. And I was thinking, as you were saying, starting your talk, you were talking about how these rhythms obviously change with age, and you also mentioned that children have difficulties forming memories. And it occurred to me that maybe as these areas of brain develop and as these theta frequencies, say in the faster range, mature, if you will, that gives them the ability to actually form those memories, and that those neural mechanisms require the theta rhythm. Do you want to comment a little bit about that?
Yeah, so let's say research from traditionally rodents, so these animal models, really say that the, memory-like tasks speak theta, and the regions that support memory, such as the hippocampus traditionally, but we'll say medial temporal lobe broadly, speak theta. And so understanding how this language, it's often how I like to think of frequencies in relation to function, how this language develops concurrently in time with the structural maturation of these regions, the functional maturation of things like the theta band, which we're showing here, and the behavioral maturation of performance over this long period. It's really a complicated picture of sort of development of structure, function, and behavior together that supports these. And so what was especially exciting about this was sort of not extending what we'd seen, as I showed from a study from the 30s, about the development of oscillations broadly, but this development does not just apply to oscillations emerging and getting faster. That was shown in Scalp Eeg with alpha. We're now showing it here as we're able to access the medial temporal lobe directly in this language that speaks memory in the medial temporal lobe with theta. As neural oscillations are also widely attributed to supporting communication, so interaction at a certain language between brain regions, to find these same pattern in the prefrontal cortex, knowing that these regions talk and that their dialogue matures, so to speak, concurrent with the memory, to find that this sort of, refinement or development or maturation of the functional language over the same period was super exciting, suggesting that something about this does very likely have to do with the development of memory. But not only that we also have, and this was a moment of true discovery, this slower oscillation going the opposite direction. So it's just about oscillations emerge, so to speak, and then get faster, about the function differentiation.
That's fascinating. That's a particularly fascinating observation. Yeah, and that's something that we will look to your lab for the future to see what more the findings that you'll get in that area. Fascinating study. Thank you.
Thank you.
And if you can unshare your screen, our next presenter is Blake Rafferty. And speaking of communication, we are going to be looking at EEG rhythms, but in communication and in language. And moving to the back end of the brain, to the left fusiform gyrus, which is one of my favorite regions of brain. So Blake, take it away.
All right, let me share my screen. All right, can you guys see that?
Yes.
Yeah, okay. Great. So yeah, hi, I'm Blake, and I'm really interested in how EEG rhythms correspond to linguistic processes, specifically syntax. So there's some recent evidence that suggests that syntax might be reflected neurologically via power increases in delta oscillations, which are a slow wave kind of oscillation. Specifically, the study by Bonage et al. In 2017 compared these sentence fragments that had some evidence of grammatical structure, like I have pictured here on the left. They were yesterday evening they compared those fragments to ungrammatical word lists, which were essentially scrambled versions of the same words. And they found that delta power was higher generally across the entire length of these grammatically structured sentence fragments. And they interpreted this to suggest that there's some type of syntactic consolidation of the individual words into unified units, which is in linguistic kind of literature referred to as unification or merge, and that higher delta power corresponded to that activity. But since syntax and language processing in general is really time sensitive, it wasn't really clear if that's what this result represented in their study, because they used a variety of different grammatical patterns in their sentence fragments. And so they were kind of forced to average across the entire sentence instead of looking at specific time points. But considering their results, we wanted to kind of build upon that by essentially studying grammar and syntactic processes and including information in the time domain. So in a previous study, we designed sentence stimuli to compare essentially word sets that formed complete grammatical units or constituents. They were little noun phrases that had like a determiner like the word the and a pseudo word like the word moop. We used pseudo words instead of real English words because it kind of limited the effect that semantics might have on the processes that we were looking at. And then we compared those little short noun phrases to the same words, but in a reversed order. And these didn't comprise any specific grammatical violations, but they were incomplete. And so they didn't form a complete constituent. And then as a control condition, we also included some familiar and unfamiliar pseudo word sequences. These essentially had no syntactic structure and no semantic information. So they kind of served as a control. And what we found was that following the noun phrases that formed complete grammatical constituents, delta power was highest, specifically after the second word, which is exactly when we would expect these kind of grammatical processes to be taking place. But we also observed increased delta power for the incomplete constituents compared to the word list. And so because these didn't really form a complete grammatical unit, we didn't think it could be this kind of unification or consolidation of words into a single kind of working memory representation, because you can't really do that to an incomplete unit like on something like moop the. But this kind of phrasal pattern does have some grammatical information. So it's possible that participants were still trying to interpret these incomplete constituents as grammatical. And so considering that, it's possible then that instead of this kind of syntactic unification process, that delta oscillations increase as participants try and build our syntactic structure as they're parsing language. But to really verify that approach, there are a couple questions that still needed to be answered. Specifically, how do delta oscillations change when sentences have multiple grammatical units? You know, a noun phrase is one thing, but when you have a lot of different moving parts, you know, do delta oscillations kind of match that? And if it does, if it represents this building of hierarchical, you know, grammatical structures, then we really should see differences at the edges of these grammatical structures rather than just across the entire sentence. So what we did is we wanted to use time frequency analysis to kind of characterize delta responses at specific time points in sentences that had these fixed grammatical patterns. So we used these fixed patterns essentially because, yeah, it let us look at specific time points. And we built 4 sets of stimuli. The first one were sentences that followed the pattern of the English cause-motion construction, which is a pretty common grammatical structure in the English language, something like the man pulled the boat down the dock. And we compared that to the same words, but in a scrambled order. And essentially that comparison, the scrambled sentences had no real semblance of syntactic structure. And in addition to that, we wanted to still control for any effects of lexicosemantic content present in the sentences. We wanted to make sure that our effect was really related to the grammatical structure. And so we modified these cause-motion constructions by replacing all of the content words, the meaningful words, with pseudo words. So instead of the man pulled the boat down the dock, we had something like the pob gurped the hink down the jarm, which grammatically is well formed, but semantically it really doesn't mean much. And we also wanted to kind of provide a similar control to the scrambled English conditions. So we created a scrambled version of the pseudo word Jabberwocky sentences that were, just scrambled. We had two different main comparisons that we were interested in. The first one was to see how grammatical structure affected delta oscillations. So to kind of explore that, we combined the English and pseudo word cause motion constructions and compared those to the combined scrambled sentence patterns. And we also wanted to see how semantic content affected delta oscillations. So for that comparison, we combined the English cause motion and English scrambled sentences and compared that to the combined pseudo word cause motion sentence and the pseudo word scrambled. So for our actual experiment, we had 23 right-handed females. I work in speech pathology, so there are primarily female students, which is why we did that. And we fitted them with an EEG cap before they entered an electromagnetically shielded booth. And after we seated them inside, we presented stimuli to them visually, one word at a time. Each word came on the screen in the middle of the screen and was there for 500 milliseconds and before it was replaced with the subsequent word. Participants were asked to read the sentences silently. And then following the sentence, there's a set of stars on the screen that essentially cued participants to press a button on a keypad that would start the next trial. And the stars that I have pictured here above the timeline correspond to essentially the points in the sentences where full constituents could be formed. So these are really the time windows that I was most interested in. All right, so here I have the results of the time frequency analyses that we did on each of these different comparisons. Here I have the delta oscillatory power at the time points that each word was on the screen. The black lines correspond to the nonsense or the pseudo word condition. The green corresponds to the English condition. And then I have the scalp distribution of that delta power pictured here across the timeline as well. There's really no large effects of lexicosemantic content here, except for after word 5, where the pseudo word condition, where there was actually no lexical or semantic information, actually caused higher delta power than the English condition. And we thought that was kind of strange. So we looked at our stimulus material and we found that it was possible that in the scrambled pseudo word condition, as well as the pseudo word condition with grammatical structure, words 4 and 5 could have formed a grammatical unit in all cases. So we think that this is kind of an effect of that, but it's impossible to say for sure. And then the comparisons that I was most interested in are the effect of grammatical structure. So the blue lines represent delta power for the grammatical sentences and the red ones represent for the scrambled sentences. And the edges of the constituents occurred at word 2, word 3, word 5, and word 8. And as the gray highlighting essentially indicates time points where the conditions were significantly different. And as you can see, the grammatical sentences were higher at all of the constituent boundaries, so all the edges of the syntactic units. And there was almost this kind of pattern of delta activity increasing until a full constituent was formed, and then a somewhat resetting that occurred when a new constituent was started. When we looked at the scalp distribution for these, they looked pretty posterior, which is not super common for, or it's not the primary areas that people tend to think of for linguistic and specifically syntactic processing. So we wanted to conduct some source analysis to identify what cortical areas are most responsible for these differences. So to do that, we used dynamic imaging of coherent sources, which is just a distributed dipole method to essentially see how power within a specific frequency range is distributed across the brain. And the results of these analysis really gave us 3 specific clusters of activity that differentiated the grammatical sentences from the ungrammatical sentences. And those were in the superior frontal gyrus, as well as the anterior portion of the middle temporal gyrus. And then the largest cluster of activity was actually focal to the fusiform gyrus, but was spread across the visual association areas. The fusiform gyrus has been suggested as this kind of interface between sensory and conceptual systems and visual processing that can be kind of used to facilitate more efficient visual processing. And so it's really common to see it activated in reading tasks like the one that we used here. In addition to this, delta oscillations coming from prefrontal areas have been suggested to exhibit top-down control mechanisms over sensory processes. And so, considering these things together, we thought that it's possible that these delta oscillations that emerge from prefrontal and visual association areas really reflect the integration of the visual sentence stimuli with the top-down knowledge that participants had with syntactic structure. So yeah, to kind of sum things up, the main findings we had were that delta power doesn't really appear to be modulated by lexical semantic content, but it does appear to increase as participants build linguistic structure and then reset after full grammatical units could be formed. And then the source results we kind of found came from prefrontal and visual association areas, which might reflect integrating the syntactic information with the incoming sentence stimuli that they had. And yeah, that's pretty much it. Thanks.
Well, thank you, Blake. And people, but don't be shy about asking questions. I don't see any questions in our chat, and I'm happy to ask questions till the cows come home.
Sure. Go ahead.
Actually, and I was gonna, and in fact, I'm going to, because Blake, I have a question, because I was thinking about when you were presenting your introduction, that how we recognize different word types. And it occurred to me that is it possible that in some way this delta kind of activity that we're seeing varies as a function of word type in some way?
It's possible. And that was part of the inspiration for us including the pseudo words, just because they didn't really have any obvious word type. So if it was sensitive to that, then we would have expected to see that difference between the English and the pseudo word comparisons. But pseudo words are obviously not a super naturalistic language stimuli. So it's possible still that in more naturalistic settings, something like a determiner like the word the and a noun might differentially affect delta oscillations. Okay,
and then since no one else is stepping up, I'm happy to ask more. When you showed your DICS source modeling, and those data looked very nice. Did you get individual MRIs in each subject when you for those?
Yeah, no, unfortunately not. Those were just a general MNI kind of MRI for us.
Okay,
we just that's projected on.
Okay, great. Thank you for that. Thank you for clarifying that.
Yeah, no problem.
Okay. Well, if there are no further questions from this very shy audience that we've got here today, thank you, Blake, for that presentation. That's absolutely fascinating work.
Thanks.
Really fascinating work. And we are going to jump back from language to memory again. And we have now we're going to move back to also alpha activity and looking at scenes and scene complexity. And that's something that's also interesting. And we're going to look at data now in kids and adolescents. And so Chin Yin is going to take us through that. So Chin, the floor is yours.
Thank you for having me. I'm Qing from Wayne State University. I'm a graduate student in Department of Psychology. And today I'm going to talk about the relationship between the maturation of the occipital cortex and memory development. So it is well known that the ventral temporal cortex has fusiform face area and parahippocampal place area. And previous studies have found that the size of functionally defined parahippocampal place area and fusiform face area increase with age. And this age-related increase in the activation area to places or faces is associated with development of category-specific recognition memory, suggesting the link between the development of high-level visual cortex and category-specific recognition memory. And the processing of low-level visual information, such as color perception, also shows developmental gains through childhood and adolescence, so probably due to the developmental changes in the occipital cortex. And it has been established that the age-related gains in memory performance are associated with development of prefrontal cortex and middle temporal lobe. However, memory for visual stimuli depends on the input from the occipital cortex. So it is unknown if the functional maturation of the occipital cortex supports the development of memory for complex visual stimuli such as scenes. So in our study, we used intracranial EEG recording in which subdural electrodes are implanted in epilepsy patients for seizure management. And thank you, Lisa, for such great introduction for the intracranial EEG recording. And it has a high temporal resolution and superior signal-to-noise ratio. So here is an example of electrode coverage of one patient. And we analyzed the data from occipital cortex from 24 participants aged 26 to 20 years old who had occipital cortex electrode coverage. And here we showed the electrode coverage on the group level, and the electrodes were color-coded by participant age. And in order to examine memory-related signal, we used a scene subsequent memory paradigm. So I'm going to just go over this paradigm pretty quick. Participants studied pictures of scenes in preparation for a recognition test. And during the test, they had to respond if they saw this picture before or not. And based on their recognition response, the studied pictures were labeled as subsequently recognized or subsequent hit or subsequent miss pictures. So by analyzing data from the encoding block, we were able to look at the occipital signal that is related to memory formation. And in order to approximate different levels of visual processing during memory encoding, we classified things as high or low complexity things defined by the unique by the number of unique object categories depicted in the scene. So as you can see here, high complexity things has more than 4 object categories, and low complexity thing has less than 4 object categories. And first, let's look at memory performance, which is measured here as recognition accuracy. And we plot age on x-axis and recognition accuracy on y-axis. Blue color represents the complexity thing recognition accuracy, and orange represents low complexity thing. And we found age by complexity interaction, which shows age-related increase in recognition accuracy for high complexity things, but not for low complexity things. And our behavioral results are consistent with the previous study in normal participants. And the memory performance results confirm that there is visual complexity effects on memory. And next, we examined if and how the occipital cortex is associated with this age-related increase in memory for complex things. So in our study, we examined alpha oscillations from the occipital cortex because they are the most prominent features in the visual regions. We examined two different aspects of alpha oscillations, the broadband alpha power and instantaneous alpha frequency. So it has been shown that the decreases in alpha power are associated with increases in neuronal activity, hemodynamic responses, and alpha power decreases indicate richness of information encoded and can predict memory outcome. And alpha frequency indicates the speed of the oscillation, and it is consistent with the notion of rhythmic perception and has been shown to relate to the temporal resolution of visual perception.
Just to stop you for a minute, Chin, there's a little bit of a problem with the sound that you've got. It's a little bit muffled, at least as I'm hearing it. So I don't know if you can turn up your volume a little bit. Sorry to interrupt you.
Oh, it's okay. So it's now better now?
I can hear it a little bit more clearly because it was just breaking up a little bit. Okay, thank you.
Okay. Okay, so now let's first look at subsequent memory effects in alpha power. Here I first show the scene viewing induced power change. So the 3 seconds of scene presentation is on x-axis and frequency is on y-axis. So the solid vertical line indicates the scene onset and the dashed vertical line indicates mean response time. So power change after scene onset is color-coded. And here we can see scene viewing induced power decreases in alpha range for high complexity heat, high complexity mist, low complexity heat, and low complexity mist. So next, we looked at if there are differences in power decreases between subsequent heat and mist pictures. So we analyzed the subsequent memory effects, which are the differences in power between heat and mist. And we found significant negative subsequent memory effects in high complexity scenes across the whole scene perception period. Here, the enclosed areas indicate the data points of significant subsequent memory effects. And few subsequent memory effects were observed in low complexity scenes. So these results demonstrate that scene viewing induced power decreases in the alpha range, and decreased alpha power benefits memory formation for complex visual scenes. And next, we looked at the complexity by age interaction effects in the alpha power because we wanted to know if there is difference in age-related changes in alpha power between high and low complexity scenes giving the observed behavioral results. So here we show the F statistic of complexity by age interaction for subsequent heat scenes. And as you can see, we observed significant complexity by age interaction in the middle of scene perception. So to unpack this interaction effect, we averaged the power within this time frequency windows for each participant and for high and low complexity scenes separately. We plotted the averaged power by participant's age. As you can see here, the complexity by age interactions were driven largely by an age-related decrease in power during the encoding of high complexity scenes, but not low complexity scenes. And next, we looked at if this age-related decrease in alpha power for high complexity scenes predicts memory performance. So we found an age by accuracy interaction for high complexity scenes in which lower alpha power predicted a greater high complexity recognition accuracy among adolescents. And this relationship was largely absent among children. And low complexity power was not related to recognition accuracy or the interaction of age and accuracy. So our results demonstrate age-related decrease in alpha power during the encoding of high complexity scenes, and also alpha power decrease is related to age-related gains in the recognition of high complexity scenes. And next, let's look at alpha frequency. Here, I first show the dynamics of instantaneous alpha frequency for high and low complexity scenes over the 3-second scene perception for subsequent heat and for subsequent mid scenes. And the 3-second scene perception, again, it is also on x-axis, and the frequency of alpha is on y-axis. Again, we wanted to know if there is difference in age-related changes in alpha frequency between high and low complexity scenes. So we show the F statistic of complexity by age interaction on alpha frequency for subsequent heat scenes. And as you can see, we observed significant complexity by age interaction in the middle of scene perception, and this time window overlaps with the time window of the same effects in alpha power. To unpack this interaction effect, we again averaged instantaneous alpha frequency within this time window for each participant and for high and low complexity scenes separately, and plotted the averaged frequency by participant's age. So as you can see, the complexity complexity by age interaction were driven largely by an age-related increase in alpha frequency during the encoding of high complexity things, but not low complexity things. And we didn't observe any accuracy effects on alpha frequency. So the results here show age-related increase in alpha frequency during the encoding of high complexity things. And taken together, we found age-related increase in memory performance for high complexity things, and we also found that age was associated with decreased alpha power and increased instantaneous alpha frequency during the encoding of subsequently recognized high complexity things. Third, we found that the decreased alpha power predicted improved recognition of high complexity things in adolescence, providing evidence linking the functional maturation of the occipital cortex to the development of memory for complex things. And these results were published on Neuropsychology. And I'd like to thank everyone who helped in this project, and I'm happy to answer questions.
Okay, thank you very much, Jin. Can you go back to your previous slide so everyone can see that paper? Aha, because I searched for that and I didn't find it. So thank you for sharing it for everybody. We have a question from Devin Kassenheiser. I hope I pronounced that name correctly. And he says, if I can ask you to speculate, do you think the power differences you observe for memory, age and accuracy reflect efficiency of memory usage or something else?
So here we so if we think memory usage, then we would link to how previous knowledge can affect how we encode the information, if I understand the question correctly. Yes, I think so. The power we decrease in occipital cortex is only in the occipital cortex, but it may be modulated by the prefrontal cortex or the medial temporal lobe, and by the top-down interactions. So if the top-down interactions may help reduce the alpha power. So I think it's possible that it's related to the memory usage. I hope I addressed this question.
He says that makes sense. Thank you for that. And so I don't see any other questions. So I've got another one. I've got a quick one. These results are fascinating, these low frequency results. And I wondered about cross-frequency coupling of these rhythms with the higher frequencies. Have you had a chance to look at what happens at some of the higher frequencies and how their power distribution relates to what happens to activity in the alpha range as it increases and decreases?
Yeah, that's a great question, and we haven't looked at that, but we are planning to look at that. So in one of our results, we found this increased frequency, and maybe this increased frequency will give more time window for like high gamma to couple with the alpha frequency. So yeah, we are following up on that analysis.
Great, thank you so much. Well, this was a great 3 set of talks, a nice hat trick. And I just wanted to say too that we have to think also too about our terminology that we use too. And so as a little advertisement, we have a document too, which encourages people to think about how they use terminology, and it's Cobitus guidelines for magnetoencephalography and EEG as well, and in fact Gary is putting those links in for people to look at, to try and encourage people to all use the same terminology, because when we go to do meta-analyses, then we can all be talking about the same things. And so I'm taking the opportunity to share that, so that particularly when we deal with activity in children and transitioning to adults, it's really hard to make those definitions. So I just thought I'd just pop that in there. So do any of the presenters have any questions for the other presenters? Because we've got a couple of minutes. They're smiling. Okay. Well, if that's the case, then I am just going to close this session and thank everybody. Thank you, all the presenters, for fascinating data sets that you took us through these complex data sets, and you did it in a very interpretable way. Thank you. And also thank you very much to Gauri Somaseka, who so competently managed our technical side on the back end as our host today. So have a good rest of the day, and I hope you enjoy the rest of the NeuroMatch conference. So thank you, everybody, and we'll see each other